Okay, so before I'm going to start talking about the Links CFDE Engagement Award, I would like to talk a little bit about Links. So first, the LINK acronym stands for the Library of Network-Based Cellular Signatures. And in general, Links has two main overarching goals. The first one is to improve our understanding of what happens to human cells when they are perturbed with drugs, other small molecules, endogenous ligands, a different microenvironment, genetic perturbations such as short herpine RNAs, CRISPR-Cas constructs, as well as viruses and other pathogens. So those response to perturbations are measured as a delta or signature, comparing the cellular state before and after the perturbation. And this is measured with transcriptomics through the L1000 assay, RNA-seq, epigenomics with GCP and ATAC-seq, proteomics via RPPA, microwesterns, P100, and mass spectrometry, as well as imaging. The second goal of Links is to create a reference library of cellular signatures where researchers can come in with their own signatures and that they collected from cells or tissues or from patients or animal models. And then they find perturbations that either mimic or reverse their input signatures using the Links library. So the Links Consortium has 6 data generation centers and 1 DCIC, which is at Mount Sinai, but there's also a group at the University of Cincinnati and Miami that are working with us. So our group at Mount Sinai is responsible for the overall administrations of the award, maintaining the linksproject.org website, the Links DCIC site, as well as developing tools like Shankar says and databases that integrate Links data with other relevant data. And we also have extensive education and outreach activities. And we also work with collaborators that are experimentalists that use links-based hypotheses and predictions about drugs and small molecules to validate them in a disease context experimentally. The two teams at Miami and Cincinnati developed Links data portal and an integrated analysis platform called iLinks. So now I'm going to go to the engagement plan with the CFDE, which is relatively straightforward, but I'm also going to talk about some other exciting things that we are currently not funded for, but I think because this is a cross-pollination meeting, I would like to really bring those up and potentially have future collaborations working on those things. So now I'm going to just quickly go through point by point to further explain these bullet points. So like Tudor showed, we plan to have a way to automatically create a manifest of metadata that describes all the links data sets and tools and convert them into a format that can be ingested into the C2M2, and as well as performing those fair shake, fair evaluations before and after ingestion. So the first shake insignia is also something that we have developed in our group, and you can, the goal is to have those insignia showing the assessments on the portal, this Links data portal, which is a new data portal that we plan to also deploy in 2021, as well as showing it on the Deriva CFDE portal. So this is just a screenshot from the Links new data portal that we have already developed, and we are still refining and want to make sure that the data there is all ready for a deployment. So one other part of our work is to move all of our resources to the cloud. And we have been really successful in developing those apps that integrate data from many resources with the Links data. And two of them are Enricher and Harmonizome, which are now already working in the cloud in the Strides account. and together they see over 30,000 unique users a month. And tools like BioJupy's and Arches 4 are more difficult to actually move to the cloud, so this is still ongoing. So now I'm going to talk a little bit about those exciting things that we are working on, not related to the CFDE project, but I think they have potential to have some integrations with the, so in this particular project, it's called an Apiter. So the Apiter, the idea behind Apiter is that we can convert Jupyter notebooks into a full stack web-based applications. So you can install this Apiter package and then insert into your Jupyter notebook those magics that then they can be compiled. So you can compile your notebook. And then when you compile your notebook, it's create this web-based application that has a form. And then once you load the form, for example, here you can load a file, you press submit, and then the notebook gets executed with the data that you upload. So far we have developed 40 of those Apiters, and also they include some analysis of links data, GText data, Kids First data. We also developing data visualization components that were mentioned in several of the presentation. And this is an example where we took the GText version 8 hard RNA-seq samples, and we're visualizing that data using UMAP together with the metadata that is available publicly. And this is within a Jupyter notebook, and you can interact with the data by changing the colors and the shape by the metadata. So hopefully I can talk to Jared and see if they might be interested in incorporating some of those visualizations into the GText portal, and also it might go into the CFDE portal. And then finally, this is something that we already talked about in the meeting, the face-to-face meeting in January at the NIH. So this is a workflow engine that takes advantage of documented open API. So we heard today that, for example, the xRNA people have converted their APIs into open API. And now what you can do, you can build those workflows on the fly. And this particular example is how you can enter, for example, a disease term, and then you do a PubMed search with that term, and then you get all the articles. And then those articles can be converted to a gene set. And then the genes that are mostly co-mentioned with the disease terms are submitted to the Links L1000 drug search engine to find drugs that maximally downregulate these genes. This is a simple example, but it demonstrates the potential and power of this approach to build those on-the-fly workflows. This is something that Shankar is also thinking along very similar lines. So finally, I just want... Oh.
Go ahead. Yeah, time is up. Go ahead.
Okay. Yes, this is the last slide. I just want to introduce you to the Links CFDE engagement team. From the top left is Sherry, Errol, Binji, and Kathleen. And on the bottom row from the left is Sherry again, Daniel, Max, and myself.